Hi everyone, I am Professor Dr. Zaliha Selamgula, NİDA Ömer Halis Demir Üniversitesi, Turkey. In this presentation, I would like to present you about the relationship of exercise and anti-aging molecules. As you know, physical activity and increased physical fitness are known to decrease the likelihood of morbidity and mortality from a variety of causes, such as reduced cardiovascular disease, insulin resistance, and hypertension, with concomitant increases in longevity. Whereas the reduction of these endpoints will necessarily increase longevity, whether physical activity also directly affects cellular aging remains unclear for either rodents or humans. Regular physical activity has anti-aging effects and reduce the risk of injuries in older individuals, but aerobic and resistance exercise training exert multiple beneficial effects on the aging body. In humans, aging impairs multiple biological processes from a molecular to organismal level and affects mental and cardiorespiratory health, body composition, and muscle function. Muscle morphology, fiber size, and functional properties, including strength, are preserved by lifelong physical activity. Regular physical activity has anti-aging effects and reduces the risk of injuries in older individuals. In cells, nicotinamide adenine dinucleotide level and telomerase activity are indispensable anti-aging factors for cellular survival. It is clear that physical activity and exercise induce the increase in nicotinamide adenine dinucleotide level and telomerase activity with anti-aging properties in the cells. So, in this presentation, we focus on the relationship of physical activity and anti-aging agents such as nicotinamide adenine dinucleotide level and telomerase activity with anti-aging properties in the cells. There is growing research interest in studying the physiological effects of nicotinamide adenine dinucleotide, given that increased nicotinamide adenine dinucleotide levels are related to an increase in longevity and nicotinamide adenine dinucleotide levels fluctuate in response to exercise and nutritional stimuli. In cells with telomerase activity, telomere length can be maintained, therefore delaying senescence and tissue aging. There are the correlation of physical activity levels with telomere length and telomerase enzyme activity. Exercise really does influence how we age, and a new study shows how regular exercise in later life can help offset the decline of nicotinamide adenine dinucleotide, an important molecule that facilitates cellular energy production inside the muscle. Nicotinamide adenine is continuously consumed as a substrate in adenosine diphosphate ribosylation, cyclization, and deacetylation, reactions that influence many physiological processes. Nicotinamide adenine and its phosphorylated form are key molecules in ubiquitous bioenergetic and cellular signaling pathways, regulating cellular metabolism and homeostasis is a promising strategy to gain many and multifaceted health benefits. So we can say nicotinamide adenine dinucleotide is necessary for life. Nicotinamide adenine dinucleotide stands for nicotinamide adenine dinucleotide. It is a molecule found in every cell in the body. It is used to power metabolism by enabling the mitochondria, the power station of the cell, to convert the food we eat into the energy our body needs to sustain all its functions. It is also carried to turn off genes implicated in accelerating aging process. It helps maintain healthy mitochondrial function, an important component of healthy hormone aging. Research shows that as we age. So, nicotinamide adenine dinucleotide has the steps age decrease, averaging a decline of 4% SD units per year. So, lifestyle choice can also influence nicotinamide adenine dinucleotide. The feasibility of other non-pharmacological strategies to increase nicotinamide adenine dinucleotide, such as following a low-protein ketogenic diet to enhance the nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide reductive redox ratio, and or intermittent fasting or regular exercise to boost caloric restriction should also be addressed. When rats were exposed to a ketogenic diet, significant increases in hippocampal nicotinamide adenine dinucleotide and reduced nicotinamide adenine dinucleotide ratio based on diverse published literature and its initial data, we suggest that increased nicotinamide adenine dinucleotide during catalytic metabolism may be a primary mechanism behind the beneficial effects of this metabolic therapy in a variety of brain disorders and in promotion, health, and longevity. Researchers from UNSW in Sydney and Harvard Medical School have discovered that replacing two naturally occurring molecules in the body could reverse symptoms of aging, potentially influencing how the body responds to and performs exercise. In a groundbreaking paper published in Cell, researchers found that a decline in the blood flow to tissues and organs with age can be reversed by restoring molecules that improve exercise capacity and physical endurance in mice. The researchers found that the two molecules could replicate the benefits of exercise, a finding that could lead to better athletic performance, improved mobility in the elderly, and the prevention of aging-associated diseases like cardiac arrest, stroke, liver failure, and dementia. For the first time, the study showed that as level of metabolic nicotinamide adenine dinucleotide decline with age, the body's capacity to exercise decreases because of fewer blood vessels and reduced blood flow. By treating mice with the nicotinamide adenine dinucleotide booster NMN and increasing levels of hydrogen sulfide, physical endurance is extended in mice by over 60%. This was the case in both young and old mice. Exercise really does influence how we age, and a new study shows how regular exercise in later life can help offset the decline of nicotinamide adenine dinucleotide and nicotinamide monophosphate, two important molecules that regulated cellular energy production in skeletal muscle. It is well known that it is possible to maintain strength and fitness until quite late in life, and that exercise can help contract typical loss of muscle mass associated with aging. However, regular fitness routines are not smoothing. Everyone does, and as a result, they may suffer from strength and mobility issues later in life. The good news is that this decline can be reversed through regular exercise, even for previously sedentary people. Exercise can restore nicotinamide adenine dinucleotide and nicotinamide phosphoribosyltransferase transferase levels in older people who take up exercise, at least in skeletal muscle. As this recent study shows, aging decreases skeletal muscle mass and strength, but aerobic and resistant exercise training maintains skeletal muscle function. Nicotinamide adenine dinucleotide is a coenzyme for ATP production and covered substrate for enzymes regulation cellular hemostasis. In skeletal muscle, nicotinamide adenine dinucleotide is mainly generated by the nicotinamide adenine dinucleotide salvage pathway in which NMPT is rate-limiting. NMPT decreases with age in human skeletal muscle, and aerobic exercise training increases NMPT levels in young men. Collectively, exercise training reverses the age-dependent decline in skeletal muscle and PT abundance, and some findings highlight the value of exercise training in ameliorating age-associated deterioration of skeletal muscle function. There is a myriad of NAD precursor supplements available, some avoiding prices, but simple exercise may offer a cheaper alternative for people willing to put in the time and effort. Even if the benefits of exercise only apply to skeletal muscle, it is worth doing to resist the age-related loss of strength and mobility. It will be interesting to see if the same holds true for other tissue types following regular exercise. If you are serious about living a long and healthy life and potentially living long enough to see the arrival of more advanced therapies that may effectively target aging, then exercise is quite simply the most reliable and cost-effective anti-aging measure that you can take right now, alongside a balanced diet and plenty of quality sleep. Telomere length is a primary biomarker of cellular aging that has recently been associated with cardiovascular disease, insulin resistance, and hypertension, and morbidity and mortality. There are correlations of physical activity levels with telomere length and telomerase enzyme activity. Telomeres are found on the ends of linear chromosomes and act as a mitotic clock, which shortens with every cell division until cellular senescence. So, telomeres are considered an important aging biomarker. Telomeres are not, however, static entities, but rather are a dynamic system. In certain cells, the ribonucleoprotein telomerase maintains and lengthens telomeres, allowing continued mitotic activity without progression to senescence. In cells with telomerase activity, telomere length can be maintained, thus delaying senescence and tissue aging. Human cells with telomerase activity include germline cells, embryonic and stem cells, and adult proliferation. Such cells suggest those found in the immune system. Recently, both telomere length and telomerase activity have been shown to be influenced by various environmental factors such as oxidative stress, psychological stress, and socioeconomic status. The researchers found that athletes diagnosed with fetogenic athlete myopathic syndrome had shorter muscle homogeneous telomere length than age and training match counterparts. Most recently, the researchers found that leisure time physical activity was positively related to leukocyte telomere length in a dose-dependent manner, providing the first evidence overall for typical physical activity in modifying telomere length. They hypothesized that greater physical activity levels would be associated with longer telomere lengths and that older men and women individuals. So, there is more anti-aging agents such as nicotinamide adenine nucleotide level and telomerase activity related studies and human trials being conducted at the moment, and hopefully in the near future there will be much more data upon which a conclusion can be formed about nicotinamide adenine dinucleotide level and telomerase activity in the context of healthy human longevity with physical activity. Thank you for listening to me.